Hello, my name is Eleazar Abraham, and I'm an undergraduate student in the Mure research group at the Department of Chemistry. The Mure lab has been studying the molecular mechanisms of the formation of quinone cofactors that are post-translationally derived from a conserved tyrosine residue. In particular, TPQ and copperamine oxidases, or CAOs, and LTQ in the lysyl oxidase or LOX family of proteins. Our hypothesis is that both TPQ and LTQ are formed from dopiquinone or DPQ intermediate via 1,4 addition of either water or epsilon amino group of lysine residue by copper and oxygen-dependent fashion. The objective of my study is to generate an LTQ-like quinone in a bacterium CAO, abbreviated as AGAO. To do so, I will use the precursor form containing no TPQ of AGAO to conduct the formation of quinone in the presence of an exogenous small amine. As you can see in the middle left of the poster are TPQ and LTQ structures, and I highlighted the precursor tyrosine in magenta. The mechanism of the formation of TPQ or biogenesis of TPQ is relatively well understood, and it is a self-processing reaction that requires only copper and oxygen. In an X-ray snapshot analysis of TPQ biogenesis by another research group, shown here in the middle of the poster, an intermediate resembling dopiquinone or DPQ was observed. Previously, we were able to trap the DPQ as an LTQ analog in AGAO by replacing aspartate 298 by lysine in the active site via site-directed mutagenesis. The X-ray crystal structure of D298K was solved, and as you can see here in the middle of the poster, a covalent bond formation between lysine 298 and DPQ was detected. In this project, we extended the idea of trapping DPQ and AGAO with an exogenous amine. Since LTQ-like quinone was formed in D298K AGAO, we decided to test a chemical rescue experiment using D298A to conduct the biogenesis reaction in the presence of an excess of N-butylamine, which mimics the lysine side chain residue, as shown in the bottom middle of the poster. We monitor the time dependent of UV spectral change shown in the right of the poster. In the absence of an exogenous amine, D298A AGAO formed TPQ with a lambda max at 457 nanometers. In the presence of an excess N-butylamine, however, we detected an LTQ-like species with a lambda max at 524 nanometers, which is 70 nanometers redshifted when compared to TPQ. After the reaction, the protein sample was spin dialyzed to remove excess amine salts. As you can see here on the right, there is no spectral change, suggesting that covalent modification has occurred during the biogenesis. In conclusion, an LTQ-like quinone was formed in the chemical rescue experiment. This strongly supports our hypothesis that DPQ is the common intermediate of both LTQ and TPQ biogenesis. In the future, we plan to study the environmental factors that support the selective formation of each cofactor. Thank you.